Most chemical species that constitute PM10 or PM2.5 are measured in nanograms, or 1/1,000,000th of 1 gram. Therefore, even small biases in chemical concentrations can greatly affect the decisions that are made with respect to source apportionment or health effects. PM10 or PM2.5 are commonly acquired on filter substrates. No chemical analysis method can adequately represent atmospheric concentrations if the filters to which these deposits are applied are improperly selected or handled. Teflon membrane and quartz fiber filters are the most commonly used for the PM2.5 and PM10 chemical analyses. In addition, cellulose fiber filters and nylon membrane filters are used to collect gaseous precursors. Etched polycarbonate membrane filters are best suited for microscopic or individual particle analyses. Most filters require prior treatment and acceptance testing of chemical background levels when the deposits they acquire are intended for chemical speciation. At least 2 filters from each lot of 100, or a minimum of 2% of filters purchased from a specified manufacturer, should be analyzed for all species to verify that pre-established specifications have been met. Lots are rejected for chemical analysis when blank levels for individual species exceed 1 microgram per 47 millimeter diameter filter. Prior to being assigned a filter ID, filters should also be individually examined for physical defects. In addition to lab blanks, field blanks should be designated and handled as normal samples. About 10% of all samples should be so designated. In addition to acceptance testing, a few of the commonly used filters require specific pre-treatment prior to sampling. Let's take a closer look at 3 of these commonly used filters: Teflon membrane, quartz fiber, and nylon membrane filters. The first type, Teflon membrane filters, tend to produce variable blank masses immediately after manufacture. As the time between manufacture and use increases, this variability decreases. Therefore, a 1-month storage period followed by a week of equilibration in the weighing environment is recommended. The temperature for equilibration must be within 2 degrees of 20 to 23 degrees Celsius, and the humidity within 5% of 30 to 40%. Each Teflon membrane filter needs to be individually examined for discoloration, pinholes, creases, separation of ring, chaff or flashing, loose material, or other defects. The filter should be rejected if it fails acceptance testing. In addition, any sets of Teflon membrane filters which exceed 2 times the X-ray fluorescence detection limits for elements are rejected. The second type, quartz fiber filters, adsorb organic vapors over time. Blank quartz fiber filters should be heated for at least 3 hours at 900 degrees Celsius to minimize organic carbon levels. After this, a sample of each batch should be tested for carbon blank levels. Any sets of filters with carbon levels exceeding 1 microgram per square centimeter are refired or rejected. Finally, all pre-fired filters should then be sealed and stored in a freezer prior to preparation for field sampling. The third type, nylon membrane filters, absorb nitric acid over time. Blank nylon membrane filters should be soaked in sodium carbonate, rinsed with deionized water, then dried in a vacuum oven. Sets of washed filters with nitrate levels exceeding 1 microgram per filter should be rejected. Pre-washed nylon filters should be sealed and refrigerated prior to preparation for field sampling. For all types of filters, the results of all filter treatments should be recorded in a database with lot numbers and filter ID numbers. In areas with large secondary inorganic aerosol contributions to PM2.5, precursor gases such as nitric acid, nitrogen dioxide, ammonia, or sulfur dioxide should also be monitored. Cellulose fiber and quartz fiber filters can be soaked in solutions of gas-absorbing chemicals prior to sampling to collect these gases. Practical use solutions consist of the following. Note that in all cases, the balance of the solution is water. A mixture of 25% citric acid and 5% glycerol solutions are used for impregnating filter substrates to collect gaseous ammonia. A mixture of 15% potassium carbonate and 5% glycerol solutions are used for impregnating filter substrates to collect gaseous sulfur dioxide. A mixture of 25% triethanolamine and 5% ethylene glycol solutions are used for impregnating filter substrates to collect gaseous nitrogen dioxide. And 5% sodium chloride solution is used for impregnating filter substrates to collect gaseous nitric acid. To impregnate the filters, filter discs are immersed in the appropriate solution for approximately 30 minutes. These discs are then removed and placed in clean Petri slides for drying in a vacuum oven. One sample from each lot of 50 impregnated filters should be extracted and analyzed prior to field sampling to ensure that filter batches have not been contaminated. Pre-treatment, use of lab and field blanks, and careful handling are only some of the procedures involved in filter handling and preparation. Along with other quality assurance techniques, they help to ensure that speciation analyses will be as accurate and valid as possible. In order to determine the chemical composition of collected PM samples, a variety of analytic techniques can be used. Since air filters contain very small particle deposits, preference is given to methods that can accommodate small sample sizes. Three commonly used analytic methods that meet these requirements are energy dispersive X-ray fluorescence, or XRF, for elemental analysis, ion analysis for major ions such as nitrate, sulfate, and ammonium, and carbon analysis for organic and elemental carbon. While there are many other analytic methods used for speciation, we will focus only on these three. First, we'll discuss X-ray fluorescence. In XRF, the filter is loaded in the cassette with the deposit side facing downward. If the filter is heavily loaded, a nylon membrane filter is first placed in the cassette, and then the heavily loaded filter is placed on top with the deposit side facing downward. A batch of 14 filters along with one replicate from a previous run and one standard are to be analyzed in each sample run of 16 filters. In XRF, the sample material collected on a filter is irradiated by high-energy X-rays, which eject inner shell electrons from the atoms of each element in the sample. When a higher energy electron drops into the vacant lower energy orbital, a fluorescent X-ray photon is released. The energy of this photon is unique to each element, and the number of photons is proportional to the concentration of the element. These concentrations are quantified by comparing photon counts for a sample with those obtained from standards of known concentration. Since emitted photons can be absorbed in a filter or heavy deposit, thin membrane filters with deposits in the range of 10 to 50 micrograms per square centimeter provide the best accuracy and precision for XRF analysis. XRF methods can be broadly divided into two categories: wavelength dispersive, which utilizes crystal diffraction for observation of fluorescent X-rays, and energy dispersive, which uses a silicon semiconductor detector. XRF methods can also be categorized by the excitation method used, direct filtered excitation or secondary target excitation. XRF is usually performed on Teflon membrane filters with an energy dispersive XRF analyzer. XRF can analyze elements between sodium and uranium on the periodic table. Some of the elements commonly found with XRF are aluminum, silicon, sulfur, chlorine, calcium, and iron, among many others. Three types of XRF standards are used for calibration, performance testing, and auditing: vacuum-deposited thin film elements and compounds, polymer films, and NIST thin glass films. When the actual analysis is done, sensitivity factors, or the number of X-ray counts per microgram per square centimeter of the element, are determined for each excitation condition. The sensitivity factors are multiplied by the net peak intensities yielded by ambient samples to obtain the microgram per square centimeter deposit for each element. The net peak intensity is obtained by subtracting background radiation, subtracting spectral interferences, and adjusting for X-ray absorption. Since the XRF method is relatively inexpensive, has low detection limits, has multi-element capabilities, and non-destructively analyzes the filter, it is one of the most commonly used elemental analysis methods. The next method we will focus on is ion analysis. Aerosol ions refer to chemical compounds which are soluble in water. The water-soluble portion of suspended particles associates itself with liquid water in the atmosphere, thereby changing the light-scattering properties of these particles. The different ion analysis methods measure, in varying ways, the reflection and absorption of light from sample particulate matter. By noting which wavelengths are absorbed or reflected, technicians can identify which ions comprise a particular sample. Noting the amount of response also allows the technicians to calculate the amount of a given ion as well. All ion analysis methods require a fraction of the filter to be sectioned, extracted in deionized water, and then filtered to remove insoluble residues. 40 to 50 filters are placed into an extraction rack, sonicated for a minimum of 60 minutes at stable low temperature, and then mechanically shaken for 60 minutes. The extracted samples need to be stored in a refrigerator overnight prior to ion analysis. The major sampling requirement for analysis of water-soluble species is that the filter material is hydrophilic, allowing water to penetrate the filter. These ion analysis methods include atomic absorption spectrophotometry, or AAS, ion chromatography, or IC, and automated colorimetry, or AC. AAS has been very useful for measuring water-soluble potassium and sodium. Simple ions such as magnesium and calcium can also be quantified by AAS. For simple ions where AAS is an effective analytic method, it is usually the first choice due to its relative simplicity and inexpensiveness. Polyatomic ions, such as sulfate, nitrate, ammonium, and phosphate, must be quantified by other methods, such as ion chromatography or automated colorimetry. Although they are different methodologies, IC and AC produce comparable results in studies. The choice between these two methods for sample analysis is dictated by sensitivity, scheduling, and cost constraints. The final analytic method we will discuss is carbon analysis. 3 classes of carbon are commonly measured in ambient aerosol samples collected on quartz fiber filters. First, organic, volatilized, or non-light absorbing carbon. Second, elemental or light absorbing carbon. And third, carbonate carbon. Sampling and analysis methods for carbonate carbon have not yet evolved for use in practical monitoring situations. For organic and elemental carbon, however, several analytic methods have been evaluated. It is important to remember, when considering these analytical methods, that the definitions of organic versus elemental carbon are operational and reflect the method and purpose of measurement. The 3 most commonly applied methods in aerosol studies for the analysis of organic and elemental carbon are the thermal optical reflectance method, or TOR, the thermal optical transmission method, or TOT, and the thermal manganese oxidation method, or TMO. The TOR method of carbon analysis utilizes volatilization and combustion to remove light-absorbent carbon while measuring the reflectance from the deposit side of the filter. Temperature is relied upon to distinguish between organic and elemental carbon. In this case, organic carbon is carbon that does not absorb light at the wavelength used, and elemental carbon is light-absorbing carbon. The TOT method applies to the same analysis method as TOR, except that transmission instead of reflectance is measured. In addition to the use of these methods, Teflon membrane and quartz fiber filters can be submitted to a light transmission measurement before and after sampling on a transmission densitometer. This method only applies to light-absorbing carbon. The TMO method uses manganese dioxide present and in contact with the sample throughout the analysis. With respect to these methods, laboratory analyses for the mass, elemental, ionic, and carbonaceous properties of suspended particles have matured. They can now be performed with commercially available instruments, following established standard operating procedures and with traceability to common standards.